Hi, everyone, and welcome to Microbial Minutes. This is the American Society for Microbiology's every couple of weekly update on what's hot in the microbial sciences. I'm Julie Wolf, the Science Communication Specialist here at American Society for Microbiology. And today is October 30th, 2018, the day before Halloween. And so I thought it would be apt to do a roundup of something quite scary, which is a disease outbreak. There have been a number of outbreaks that have been ongoing in the last weeks and months, and we only update on these every once in a while. So I thought today what we would do is to talk about these scary outbreaks, and this would be the three that we are going to concentrate on are the acute flaccid paralysis, which has been in the news, a drug-resistant salmonella strain, which has been identified in chicken, and the Ebola outbreak that has been ongoing in the Democratic Republic of the Congo. Then to ease away from the outbreak nature, we're going to talk about something a little more positive, which is sequencing tools that may aid precision diagnostics at the very end. So let's dive right in. You may have heard about acute flaccid paralysis. It's been in the news quite a bit lately. And our first report from MBio focuses on one of the possible culprits of acute flaccid paralysis. The take-home message of this report is that enterovirus D68 infects human neurons and causes mouse neuropathy. I should really say neuropathogenesis, but it didn't fit on the screen. And so in this article, we're going to talk about acute flaccid myelitis, also called acute flaccid paralysis or flaccid paralysis. This is a condition where patients have very weak limbs. Sometimes that can also culminate in a facial droop because the facial muscles will stop holding the same position. And it comes on quite suddenly. This is something that can be either temporary or long lasting, with patients requiring long term care, and has so far affected mostly children between 1 and 18 years. So that's quite a range. The average is somewhere around 10, but really children of all ages have been affected by this very mysterious condition. Now, this might sound a little bit familiar to you. This is very similar to the condition that was caused by poliovirus. And poliovirus was an enteric virus that was fecal oral, meaning, and it is, I mean, it still exists in certain parts of the world. We don't experience it here in the United States, but it was, when it was in the US, it was passed fecal oral, and so contaminated water could be a potential source. This is one of the reasons that pools were closed in the summer, so that if there was a polio outbreak, they would close the pool so that there wouldn't get any contamination in that pool that could, even though you're not drinking it, could potentially infect an individual. Now, there's a number of potential candidate viruses that might be associated with the current outbreaks. And the one that we're going to discuss today is a candidate called enterovirus D68 or EVD68. This is also an enterovirus. It's related to poliovirus. Both of these are picorna viruses, or as I like to think of them, pico-RNA viruses, because they have small RNA genomes. And there's a number of different viruses that fall into this picornavirus group. You can think the polioviruses, enterovirus D68, and another one you may have heard of, enterovirus A71. Both of these are picornaviruses. Rhinoviruses are also picornaviruses, and rhinoviruses are the virus that is associated with the common cold. So this is a respiratory virus. So it doesn't have to be something that's passed fecal orally. And that's important to note because this enterovirus D68, although being called enterovirus, and you might think has to do with the enteric system, it's actually a respiratory pathogen. It causes respiratory disease in children. Now, in some cases, it may cause an asymptomatic infection, and it can cause all varying degrees of disease. It can be severe enough that children will go to the hospital or to their healthcare provider, and that's where some of the data about EVD68 epidemiology has come from, when people actually present at the healthcare facility and are tested for what virus might be causing that disease. From that type of research, from epidemiological studies, we know that there was a major outbreak of enterovirus D68 in 2014. There seems to be a resurgence and an outbreak of enterovirus D68 every 2 years since. And when people looked even a little more historically, it looked like there was a little bit of enterovirus being passed around in 2012 as well. And this is the first piece of evidence that suggests that enterovirus D68 might be associated with acute flaccid myelitis, these weak limbs and facial droop symptoms. And so what I'm showing you on this right-hand side graph, this is not the enterovirus D68 cases, but the number of acute flaccid myelitis cases, so the number of children that are presenting with this flaccid paralysis. And you can see in 2014, 2016, and now in 2018, once again, we're having a resurgence in the number of cases, just as cases of enterovirus D68 have this every 2-year resurgence. Of course, this is just a correlation. You can probably find other patterns that happen every 2 years. And so this more recent publication from MBio lends support to enterovirus as a potential cause of this acute flaccid myelitis. So in this case, we're going to look at data from a couple of different enterovirus strains. There's a couple different circulating strains. They have different genetic mutations and behave just a little bit differently. One of them is clade A, and in the research paper, this is going to be the isolate from Tennessee, which on the right-hand side is demarked in blue. And one of them is clade B. This is from Missouri, and in this case is demarked in red. Now, those are both currently circulating. Enterovirus itself was identified as a respiratory pathogen in 1962, and the authors also examined the activity of a historical isolate. This wasn't, I don't believe, the actual 1962 isolate, but it was something that was more like what had originally been identified decades ago. And so this is the VR1197, which is demarked in green. The first thing that the authors wanted to ask is there a difference in these strains between human neuronal cell infection? So the ability to infect human cells. They looked at the ability of all of these different viruses to infect HeLa cells. You may be familiar with HeLa cells. They're an epithelial type of cell that is used for a number of different cell assays. All of the different strains could infect, replicate, and produce progeny viruses within HeLa cells, but only clade B, the Missouri strain, could replicate, could both get into and replicate and make progeny viruses inside a human neuronal stem cell line. And what they wanted to ask after that, after determining that the clade B virus could replicate in human neuronal cells, was to ask whether there was any association with disease. Now, you can do this by looking at people and doing epidemiological studies, but in this case, they were looking in mice. They infected mice with one of these 3 strains, and this is what the first graph on the top here is looking at the motor impairment. So how much problems the mice had moving their limbs. In this case, a higher score along the x-axis indicates more impairment or a worse worse course of disease. And you can see only one of these strains is able to cause this motor impairment. And again, we're looking at the clade B strain, the one from Missouri, whereas the other current circulating strain from Tennessee, this clade A strain, has no motor impairment, and neither does a historical one. When the scientists then looked at the mice to see whether that virus had actually crossed into the central nervous system, only that clade B virus was able to be propagated, was able to be found within that spinal cord fluid, whereas all of the different viruses were able to propagate and make new viruses within muscle cells, the muscle tissue of the mice. The researchers then wanted to look at the relatedness of different circulating strains. So they took sequence data that's available and they made a phylogenetic tree looking at the relatedness between a number of the different enterovirus D68 clades. You can see here on the right-hand side, there's a whole lot of data, right? You can see all the clades which are related by similar sequences, and then you have additional data overlying this. So we have the cases that have been associated with acute flaccid myelitis in green. So those are things that have caused, those are strains that have caused human disease. We have those that were tested in this case for the growth within neuronal cells. You can see the pink arrows shown here. Those that were not able to grow in the human cells are called SH cells. You can see a few were not able to up here and up here. And the paralytogenic, or those that were able to cause that neuropathogenesis in mice, are demarked in blue. So here, you can see almost every place that there is a pink arrow, the ability to grow in human neurons, you can see that there's also a blue arrow, which is indicative of the ability to cause disease, this neuropathogenesis in mice. And so what the authors suggest is that because of the similarity between disease in mice and growth in human neuronal cells, this ability of the viruses to grow in human neuronal cells may be a good proxy assay for neuropathogenesis. It might be one way to look at the potential of different strains that are circulating or isolates that are taken from patients to look at their abilities to cause this neuropathological disease. And so this is something that adds to the possibility of enterovirus D68 as one of the causes, or perhaps the cause, of acute flaccid myelitis. Now, this acute flaccid myelitis has been making headlines in a number of different places. This has made headline news. I even had my mom on the phone yesterday asking me what's going on with these children who are becoming paralyzed. And this is a medical mystery that is of great concern. Of course, when something like this happens, especially to children, and there's no known disease cause or etiology, there's a lot of investigations, and this is something that's being prioritized right now by the CDC to identify whether there is a causative relationship between this particular virus and the condition. This is not this particular study adds again a layer of support, but does not show that the enterovirus D68 causes this same human disease. And one of the things that I thought explained in a mainstream media most clearly about the difficulty in finding what the cause of this is this piece from NBC News by Maggie Fox, which had several different sections that outlined why it's so hard to associate one particular virus or one particular disease with these symptoms. And I mean, first, one considers the rarity of the cases and considers that when you have a respiratory disease, it's very rare. It has to be quite severe for people to actually go to the doctor. And even then, for the doctor to take a sample and say, okay, well, we're going to sequence this and figure out what virus it is, instead of simply saying, okay, you have a virus, just go home and rest and it should take care of itself. Of course, often by the time patients have developed this particular symptom, they have the weak limbs, the facial droop, the patients may have cleared the virus. So you might not be able to sequence that virus anymore. And I think one of the most complicating conditions here is that there are many different viruses that can cause this acute flaccid myelitis or this flaccid paralysis, or that are associated with different neurological symptoms. And tying a single one to all of these cases might prove difficult. It's possible that there are a number of different viruses that can cause this particular symptom. So we will stay on top of this in Microbial Minutes. Tune back for future updates. We're going to move on to our next outbreak, which is Salmonella. So this is an investigation notice that was posted on the CDC's website. It's titled Outbreak of Multi-Drug Resistant Salmonella Infections Linked to Raw Chicken Products. Take-home message is that drug-resistant Salmonella infantis is reported in chickens. So this is a new Salmonella outbreak strain, which has been identified as Salmonella infantis. And I'm putting new in quotation marks there because you can see by the timeline on the right-hand side that there have been cases reported of this strain causing illness in people for months. This has been ongoing. This is an ongoing outbreak that has had people getting sick from this same strain, which they've identified as the same strain through sequencing techniques, since early this year, since January of 2018. And it's been pretty consistent with reports coming in just about every week with another individual or maybe another two individuals who have been affected. And of course, those that present again at the hospital are the ones that are representative of a much larger population of people who feel a little bit of GI distress and perhaps take a day off from work and just rest and eventually feel better on their own. So this has been tied to chicken, but there has not yet been a single supplier that has been pinpointed as the source of that contaminated chicken. In fact, the widespread nature of different types of patients that have come into the healthcare facilities suggests that it might not be a single supplier, since there are some people who have reported being sick after going to a restaurant, some people who have made food at their own home, wholesalers. So this means that it might be a farm that's providing chickens to a number of different suppliers and not necessarily a single supplier. These cases have been reported in 29 different states. There have been 92 people who've been reported as sick, 21 hospitalizations, and fortunately, as of yet, no deaths. And one of the reasons why this particular strain is being highlighted is because it is resistant to a whole host of different antimicrobial agents that attack the microbe in a number of different ways. And so, um, should this strain gain a few more genetic determinants that allow it to resist the few remaining drugs that are still active against this strain, it could be very dangerous, which is one of the reasons why the CDC, of course, is trying to quash this outbreak. Fortunately, there are still a couple of drugs that are effective against this Salmonella outbreak, and we will update you should this change. This has been covered in a couple of mainstream outlets. It's always good to let the general public know when there's an outbreak going on so that people take extra care. Of course, you should always make sure to not only wash your hands and cook your food thoroughly, but also to wash any food prep items, including things like the cutting board that you might use. And one of the items that the CDC has been including in their news briefs has been to make sure to not feed any of undercooked or raw meat to your pets, since they can also suffer from the disease from salmonella. All right, moving on to our next outbreak. We are now going to talk about a statement released by the World Health Organization on the October 2018 meeting of the IHR Emergency Committee on the Ebola virus disease outbreak that's ongoing in the Democratic Republic of the Congo. And so the take home message from this brief is that the WHO, the WHO, says Ebola is not an international public health threat. So before we get into this meeting and what happened there, I'm going to go over some of the numbers for this outbreak that's been ongoing for a while now, for a number of months. So if you go to the World Health Organization's website, you can find these numbers for yourself. They have an entire page dedicated to Ebola that I have screenshotted here. If you look down here on the lower left-hand corner, there's this latest numbers from the DRC. And so if we go there, we can see that the total cases so far have been 267 of those confirmed, meaning not simply displaying the symptoms of Ebola and having been in contact with someone who has Ebola, but actually confirmed clinically, 232. There have been 170 deaths, with 135 of those confirmed to be due to Ebola. So it's 267 cases since August 1st. Remember, the DRC had an outbreak in July as well, which was able to be quashed quite quickly or quelled due to the efforts of a number of different outlets, of different healthcare organizers trying to not only treat the sick, but also to use um, a new and experimental vaccine. So that this may sound a little bit familiar if you've listened to previous Microbial Minutes. We covered a number of different Ebola virus vaccines. And in the current outbreak, there's more than 20,000. I read nearly 30,000 people recently who have been covered by this particular vaccine. The vaccine, as a reminder of how this vaccine works, the glycoprotein from Ebola virus, shown in red here, is expressed in VSV, which is a non-human pathogenic virus. And so the patients or the people who receive this vaccine will experience the glycoprotein of Ebola, but without the disease, and they should create antibodies that will hopefully block that Ebola virus from being able to enter their cells should they encounter the Ebola virus. Of course, it takes a number of weeks for that memory to be formed within an individual after vaccination. But at a recently presented at a conference just recently, the group who is studying the effect of these different vaccines said that they now have good data suggesting that this vaccine, so this particular vaccine that I'm showing you here, is one that's licensed. I'm sorry, it's being developed by Merck, but it confers protection for a good 2.5 years. This has been administered in a ring vaccine style, meaning that people who are sick have their close contacts identified, and family members, people who they may come into contact with, their neighbors, people who are most likely to get sick are the ones who have been administered this particular vaccine. And that includes healthcare providers as well, who, of course, are interacting with many people who have this disease. However, despite despite the coverage of this particular vaccine, the numbers are continuing to rise. Yet, despite that, at this recent meeting in October, the World Health Organization Committee decided that a public health emergency of international concern, or PHEIC, should not be declared at this time. So, what's a PHEIC? This is an announcement that will embolden the resources of the world to be centered in order to quash or to stop a particular outbreak before it grows even further out of hand. This has only been announced a handful of times. The most recent PHEIC was in 2016 when the Zika virus outbreak was ongoing. Ebola has deserved a PHEIC announcement or declaration before in the 2000 Ebola virus outbreak in West Africa. But it's also news when the World Health Organization declares something not as a public health emergency of international concern, such as in the 2013 case of the Middle East Respiratory Syndrome coronavirus, or MERS coronavirus outbreak, which was garnering a lot of attention in the Middle East, because this will determine a lot of the resources that are dedicated or not dedicated to a particular region. The committee did highlight the very complex security situation. And so this is something I want to discuss because this is one of the reasons why this outbreak is not ending as quickly as the previous DRC outbreak earlier this year was able to be contained. Just in the last couple of days, there have been a number of headlines highlighting the very dangerous nature for citizens, civilians, and people who are in this particular area of the DRC. There's a number of different factions who are fighting, and that this has spilled over into the communities where Ebola is affecting people. And so this is the disease itself is increasing in the places that are most associated with this fighting. Unfortunately, to the detriment of everyone. For example, the healthcare workers themselves are actually attacked frequently. As you can see in this release from the Associated Press, healthcare workers in Congo's Ebola outbreak attacked weekly, and this is to the detriment, of course, to the people who are attacking them as well. Not only are these healthcare workers not able to administer vaccines and treatments to people who are sick, but this will increase the exposure of the attackers to potential Ebola patients. And this it does have negative effects. For example, as highlighted in this story, a group of youths in Beni recently stole the body of an Ebola victim on the way to the cemetery. After touching a corpse, one of the youths became infected and died. And so this is one of the reasons why this particular outbreak is not being contained as quickly as the one earlier this year was. And for more current updates, as always, I encourage you to follow Helen Branswell on Twitter, who is going through the numbers on an almost daily basis and putting them into context. She's followed Ebola among other infectious disease outbreaks for many, many years. And in her opinion, based on the numbers that have been reported thus far in October, as she states here, this is going to be a big outbreak. And so hopefully we will not have too many more updates about this particular outbreak if it's able to be contained. But should it not be able to, then we will cover this again in future Microbial Minutes. Moving on to our last topic, we're going to talk about two different reports that came out that should help to bring sequencing data into the clinical microbiology lab. One is from Nature Medicine, titled Precision Identification of Diverse Bloodstream Pathogens in the Gut Microbiome, and the other is from MBio, titled BACCAP-seq, a platform for diagnosis and characterization of bacterial infections. Take-home message from both of these reports is that new tools will help better characterize bacterial infections. And this is particularly relevant for bloodstream infections or sepsis cases, which need to be treated as quickly as possible, as soon as possible, because bloodstream infections can not only cause massive inflammation, but can seed infections throughout the entire body. The vascular system is basically the super highway throughout the body that the microbes can ride and then exit and cause infection throughout the entire system. And so this can very, very quickly lead to bad consequences for patients if they are not treated with antibiotics very quickly. One of the ways that people look at what the causative agent of these different infections is now is by culturing, of course, and sequencing. There's sequencing that is done within the lab, but uh, the ability to sequence and to gather information in sequencing has been moving forward so quickly that the tools to apply this in the clinic have been lagging behind a little bit. And so these are two different tools that may help target the search when looking for a candidate cause of a bloodstream infection that will not only help to identify the agent that is causing that disease, but can also characterize certain virulence traits, for example, whether a toxin is present within the causative strain, or whether there are particular genes that are associated with antibiotic resistance that might influence the course of treatment. So we're going to talk first about the Nature Medicine piece. This is a piece that introduces Strain Sifter. Strain Sifter is a streamlined bioinformatics tool which will match bloodstream pathogens and their genetic sequences precisely to a candidate source. So what this does is to take the genetic sequence of a bloodstream infection isolate and then compare that against the metagenomic sequences of a candidate source. And in this case, they were looking at patient stool because there are many different fecal members of our microbiome that are fine if they are in our microbiome, but of course, if they get into our bloodstream, can cause a serious disease. So what they did was to match gut microbiota to bloodstream infection patient samples. And they did this in a cohort of people who were undergoing, I believe, bone marrow transplant or were otherwise immunocompromised. And so what they found was that, as one might expect, patients who have infections with E. coli and Klebsiella pneumonia, these bacteria that are associated with the microbiome, have concomitant gut colonization by the same strains, suggesting that the source of infection was the patient themselves. Now, this could be from a tear in the intestine. It could be from just one or two of those microbes getting into a cut in some way, but that has somehow seeded. It was a self-seeded infection. But what they were surprised to see was that there were cases of Pseudomonas aeruginosa or Staphylococcus epidermidis that were also associated. These were the cause of the bloodstream infection, but they were also found in the patient gut. Now, often when a patient has a Pseudomonas aeruginosa infection, the hospital staff may assume that has to do with an environmental um, uh, contamination, and perhaps the infection control policy needs to be changed. But what this suggests is that even in these cases of environmental and skin sourced infections, things that are often thought to come from different places, they might be existent within that patient's gut. And so we can't make assumptions based solely on the identity of the isolate for where it from whence it came. Now, the second article here from MBio introduces BACCAPSeq. Now, this is a targeted sequencing platform that tests for all of the known human pathogenic bacteria. So that's 307 pathogenic bacteria, and it tests for them by incorporating 4.2 million overlapping probes. And these overlapping probes are associated with magnets. So the probes can be incubated with whatever target sample they may be interested in testing. A patient's blood sample, which will of course also have host DNA as well as bacterial DNA. When those probes are incubated with the patient's isolate, the probes that are identical for whatever the causative agent is will hybridize and then can be pulled out and sequenced, and information can be gathered about what the nearby sequences are that's next to those probes. What this has done is improve the sensitivity of sequencing methods 100 or even 1,000 times fold. And so in this case, what I'm showing you on the right-hand side is the Klebsiella pneumonia genome, which is a circular genome, but we're showing it in linear fashion here. And the number of hits, or the depth of reads, is indicated in blue by the BAC-CAPS-seq targeted sequencing, or in brown with the unbiased high throughput sequencing. And you can see that's almost down here at the bottom. It's hardly higher than the line itself, whereas we would get many, many fold times higher reads by the BAC-CAPS-seq targeted method. And in fact, the targeted method is targeted in such a way that they are interrogating the open reading frames, or the protein coding sections of the genome, which is one of the reasons why you see gaps in the knowledge here. But this allows the researchers to identify genes such as toxin genes, as I mentioned, or genes that are associated with bacterial resistance, such as the BLA-KPC gene, which is associated with penicillin resistance. And so this will give information both about the identity of the bacterial species, as well as different characteristics, which will be important for the ability to treat a patient with the correct drug and very quickly. So this was highlighted, both of these were highlighted in various places. The NIH director, Francis Collins, highlighted the first Nature Microbiology, Nature Medicine piece, stating how difficult this actually is. So the concept is simple: to find a match for the blood isolate with the potential isolate of where it may be. But of course, sifting through all of that microbiome data in order to find that match is not easy. And so it does represent a lot of hard work and a simplification of a very complex process. And as Collins writes, the findings suggest that rather than catching an infection from the environment or another person, many of the patients were becoming ill due to a microbial imbalance within their own bodies. This is talking about those Pseudomonas or Staphylococcus gut microbes that were present within the microbiome, whereas they had previously been presumed to be from other sources. One of the lead scientists from the BAC-CAPS-seq, Ian Lipkin, spoke and gave a quote about the use of the BAC-CAPS-seq, not only in clinical diagnostics, but as Lipkin says, as the sensitivity of BAC-CAPS-seq became apparent, we realized it could be used as a discovery tool for identifying biomarkers for antibiotic resistance that were expressed early after exposure to antibiotics in culture. And we hope that others will find these data useful for basic and applied bacteriology. So Lipkin and the team of researchers are hoping that this BAC-CAPS-seq targeted sequencing will help people not only in the clinic, but also for basic discovery research. This study was also interestingly picked up by the Daily Mail. It's always interesting to see what the Daily Mail picks up and decides is important to highlight for their readers, since this is a very tabloidy type of outlet over in the UK. But they did pick up the highlighted text, the same thing that I talked about earlier, saying that this new sepsis blood test is 1,000 times more effective at picking up on the deadly condition than existing methods. So that's going to wrap up our outbreak special. So the important things that have happened in the microbial sciences that we discussed today is that EV, or enterovirus D68, infects human neurons, causes mouth neuropathogenesis. We talked about how drug-resistant Salmonella infantis is reported in chickens, and this is an extremely drug-resistant isolate. We talked about the WHO and how it has said that Ebola is not an international public health threat. And finally, we discussed some new tools that will help better characterize bacterial infections. I hope you enjoyed this Microbial Minutes. If you have suggestions going forward, feel free to leave them in the comments or let us know. All right, I'll talk with you next time on Microbial Minutes.